Hello everyone, welcome to C Academy YouTube channel. My name is Velileni Ngosi. In this lesson, we will discuss the examination guideline regarding the DNA, a code of life. By the end of this lesson, you will know the secret that the examiner wants you to know in DNA, the code of life, before you sit for an examination. This document was downloaded from the Department of Basic Education website. Without any further ado, let me reveal the secrets. Secret number 1. Revision of the structure of the cell with an emphasis on the ribosome, cytoplasm, part of the nucleus. So, you remember in grade 10, we studied plant cell and the animal cell. So, in grade 12, we will focus on cytoplasm, ribosome, and the nucleus. What you must know here, you must know the functions of the cytoplasm, functions of the ribosome, and the function of the nucleus. So, and you must be able to label the structures of a cell. The secret number 2 that the examiner wants us to know is to know that there are 2 types of nucleic acid. Namely, we've got DNA and the RNA. DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. So, you must know that in a cell, we've got 2 types of nucleic acid. And then number 3, according to the examination document, we must know that nucleic acid consists of nucleotides. Let me show you the structure of a nucleotide. This is a nucleotide. If you link nucleotides together, you end up with nucleic acid. So a nucleotide is the structure like this one. Then now we can go to number 4. Number 4, according to the examination guideline, number 4 say we must know the location of the DNA. So there is a DNA present in the nucleus. So the DNA that is present in the nucleus of a cell is called nuclear DNA. So, and then another DNA is present in a mitochondria. And that DNA, we call it mitochondrial DNA. And then we've got another DNA that is present only in a plant cell. So the DNA that is present in a chloroplast, we call it chloroplastic DNA. And another thing that we must know, like the secret number 5 that the examiner wants us to know, we must know the brief history of the discovery of the DNA molecule. So we must know who discovered the DNA. So any textbook of grade 12 life science, there is a brief history about the discovery of DNA. And then the secret number 6, we must know the structure of the DNA. So the natural shape of the DNA molecule is a double helix. And then each strand of the helix is made up of a sequence of a DNA nucleotide. So here, this is the structure of a DNA. The structure of a DNA is double helix, double helical strand. If you can see, we've got 2 strands here, which are helical. Helical means it's almost like a spring, like a coil spring. And then here, I've showed you how the nucleotides are put together to make the DNA molecule. We've got a nucleotide here. This is a nucleotide, this one. And then we've got another one, and then on the other side, we've got another nucleotide. Then another one. So these nucleotides are linked together to form one strand of a DNA. And then on the other side, we've got another nucleotide which form another strand of a DNA. So these strands are held together by weak hydrogen bond. The bond that is in between nitrogenous base is called weak hydrogen bond. So this is another information that you must keep it in mind. And then we go to secret number 7. So number 7 say there are 3 components of a DNA nucleotide. We have nitrogenous base, we have sugar portion, and then we have phosphate portion. So here the nitrogenous base is linked by weak hydrogen bond. Like I've showed you, the bond between the nitrogenous base is called weak hydrogen bond. And then there are 4 nitrogenous bases, which is called adenine, thymine, cytosine, and guanine. Those nitrogenous bases, they have a sequence of pairing. Pairing of the bases in DNA occurs as follows. A pairs with T. This means adenine pairs with thymine, while cytosine pairs with guanine. This is how they pair. And then the sugar portion of a DNA nucleotide is called deoxyribose. So let me show you again. So here is this is the phosphate group, and then here we've got sugar molecule, and then this is the nitrogenous base. So the nitrogenous base is divided into 4. We've got adenine, we've got thymine, we've got guanine, we've got cytosine. So adenine pairs with thymine, while guanine pairs with cytosine. This is how they pair. So they held together by weak hydrogen bond. And then another thing that the examiner wants us to know is the function of the DNA. So the function of the DNA makes up genes which carries hereditary information. So this statement, hereditary information is an information that is from a parent and passes to their offspring. For example, if my parents, like my father, have a blue eye and I have a blue eye, this information is passes through the DNA in the form of genes. So this is the function of a DNA. Another function of a DNA, the DNA contains coded information for protein synthesis. By the protein synthesis, we mean the production of the protein or the formation of the protein. In a cell, there is a large number of protein that are produced. So before a protein is produced, the information must be carried from the DNA. Then that information is used to produce that protein. So another function of the DNA is contain the coded information for protein formation. And then the secret number 9, we must know the process of DNA replication. So DNA replication is the process through which DNA makes an identical copy of itself. So if you see in the structure, at first we have one DNA. So, and then the DNA replicate, we ended up with 2 DNA. If the replication goes until, we will end up having 2 DNA. So this is the structure that you must be familiar with when they talk about DNA replication. This is the replication, so you must know the process. So let's see what they say. What did the document say about DNA replication? You must know the thing that we must know. When in the cell cycle is the replication takes place. So let me show you the cell cycle. This is the basic structure of a cell cycle. We've got the M phase, we've got the interphase, and then interphase is divided into phases. We've got G1, which is growth phase or gap 1, and then we have S phase, which we call synthesis phase, and we have G2. which is gap 2. So DNA replication happen during the interphase. Specifically, it happened during the S phase. This is where the duplication happened. So you will find this question maybe in multiple choice or in one term. They will ask you, when is the DNA replication happen during the cell cycle? So you just say the interphase. And then another thing about the DNA replication is where in the cell it takes place. You must know where in the cell is the replication takes place. So DNA replication takes place in the nucleus. It happens in the nucleus of a cell. And then how the DNA replication takes place. So here you have to explain. You have to explain the explanation. I will make a video for each topic here. Then obviously I will make one for DNA replication. And then when you explain the DNA replication, names of the enzyme are not required. You just say enzymes. So their name are not necessary. And then another thing about DNA replication, you must know the significance of a DNA replication. Significance means the importance. You must know the importance why DNA replication takes place. So now we go to number 10. Secret number 10, interpretation of the DNA profile. Uses of the DNA profile. So this is the profile. This is DNA profile, DNA profile, DNA profile. So it can be used to identify a criminal if maybe someone committed a crime somewhere and then their DNAs were found in the crime scene. Or it can be used to find the siblings of someone, because this is the information that is found in a DNA. So what they do, they take someone's DNA and then they take another DNA of someone who they suspect maybe they share the same DNA, then they compare. If these lines are lining like this profile 1 and 2, that is mean the chances of having the same DNA are very high. But if these lines are not matching, so that is mean those people do not have the same DNA. So this is the purpose of a DNA profiling. So everything is in the textbook. So now we are going to RNA, ribonucleic acid. So what do you must know about RNA? You must know the location of the RNA. We have mRNA, which is messenger RNA. Messenger RNA is formed in the nucleus and functions on the ribosome. And then we have tRNA, is located in the cytoplasm. Let me show you this sketch. In this sketch, we've got, this is a nucleus. And then if this is a nucleus, then this space is a cytoplasm. So an mRNA is formed in the nucleus. It takes the information from the DNA, then moves out of the nucleus via nucleopore, and then functions in the cytoplasm or on the ribosome. And then tRNA, you'll only find it in the cytoplasm. This one you'll only find it in the cytoplasm. mRNA is produced in the nucleus and then moves out of the nucleus via nucleopore, then functions in the cytoplasm or on the ribosome. This is what the document is telling us. And then number 2, the structure of an RNA. You must know the structure of an RNA. RNA is a single-stranded molecule consist of nucleotides. So here is a simple structure of an RNA. You remember DNA is double-stranded. RNA is single-stranded. That is the difference. And also RNA is made up of nucleotides. Then it contains phosphate group, contain nitrogenous base, contains ribose sugar. The sugar of the that we found in an RNA is called ribose sugar. Okay. And then we go to number 3. So number 3, three components of an RNA nucleotide. So I've just explained them. The component of a nucleotide, we've got nitrogenous bases, we've got sugar portion, which we call it ribose sugar, and then we've got phosphate portion. So nitrogenous bases of an RNA, there is a small difference of nitrogenous bases from DNA and RNA. Both contains 4 nitrogenous bases, but with RNA, we've got adenine, we've got uracil, we've got cytosine, and we've got guanine. So instead of thymine, RNA is replaced with uracil. So that is the difference between, another difference between DNA and the RNA. One contain thymine and another contain uracil. So another difference is the sugar molecule or the sugar portion. The sugar portion of an RNA is ribose sugar, while the sugar portion of a DNA is deoxyribose sugar. This is another difference between RNA and then DNA. So I can show you here that the RNA nucleotide it contains phosphate group, contains sugar molecule, which is ribose, and then it contains nitrogenous base. So the nitrogenous base of a RNA is adenine, uracil, guanine, and cytosine. So we don't have a thymine here. Then now we go to the functions of the RNA. RNA plays a role in the protein synthesis, like the sketch that I've showed you. We've got RNA that is formed inside the nucleus and then move out of the nucleus. And then we've got another tRNA that is forming proteins. This is the formation of a protein. And then now we go to the process of protein synthesis. Protein synthesis, like I said, is a formation of protein. So protein synthesis is divided into 2 phases. We've got transcription, which takes place in the nucleus, and then we've got translation. Translation takes place in the cytoplasm. So to show you the example, when the mRNA takes the information from the DNA, that process is called transcription. And then it's a process that you must be able to explain it. So you will be asked during the examination, say, explain the transcription, then you must explain it. And then we've got translation. The translation is when tRNA that carrying amino acid bond with mRNA with the help of ribosomes, then to produce the proteins. So transcription takes place in the nucleus, translation takes place in a cytoplasm. Number 6, you must note the involvement of a DNA and RNA in protein synthesis. So you must know that DNA and RNA, what are they doing to produce protein? So what happened is RNA takes the information from the DNA, then moves out of the nucleus. A specific protein is formed with the information from the DNA. Then number 7, you must note symbol diagram to illustrate transcription and the translation in a protein synthesis. This is the symbol diagram. This diagram represents the protein synthesis. So you must know how to label it, and then some of the functions of this part, you must know them. Like in mRNA, 3 adjacent nucleotides are called codons. So it's very important. this one again. And then this one, tRNA, contains 3 nucleotides. They are called anticodons, while tRNA carries amino acid. So you must know again, this is amino acid, tRNA, and then these nucleotides are called anticodon. And then 3 adjacent nucleotides are called codon. And then ribosome is used to bond tRNA and the mRNA. This is a ribosome, this one. So it's used to bond them. So, and you must know how this is nucleopore, is where the stuff in the nucleus used to exit. Yeah, this is what the examiner wants us to know. So if you find this information very helpful, please subscribe to our YouTube channel. Then I will appreciate if you subscribe and like and comment. So everyone who is studying, I say good luck with your studies. Thank you.